Hi, this is Dr. Neela Bakore, and in the previous segment we started discussing the various methods of reproduction, and we finished all the methods of asexual reproduction which are seen in organisms. Now, in this particular segment we are talking about some basics of sexual reproduction shown by organisms, and then we will also see a different method of reproduction which is not common to all, called parthenogenesis. So, let us first talk of sexual reproduction. Sexual reproduction is when the gametes are formed and they fuse. So, this involves formation and fusion of gametes, and these gametes are formed as a result of meiosis or reductional division. Normally these gametes come from two parents, one male and other female, but in certain hermaphrodites like tapeworm or Taenia solium, both the sex organs are present in the same organism. So, its testes produce sperms, its ovaries produce the eggs. So, gametes are formed, but they are not coming from two individuals or two parents. So, normally whenever we talk of sexual reproduction, we say it is biparental. So, here we will write it is biparental with exceptions of hermaphrodites. Exception is hermaphrodites. Hermaphrodites are those organisms in which one individual has both the sex organs, ovaries as well as testes. Taenia solium or tapeworm is the example. In such hermaphrodites, also meiosis takes place, gametes are formed, they will fuse, they also favor cross fertilization, but the gametes are coming from one individual. So, biparental term is not applicable for them. So, except these hermaphrodites, in all other cases it is biparental. In sexual reproduction, because meiosis takes place which involves crossing over, variations are possible or variations are seen. So, in sexual reproduction, variations due to crossing over are seen. So, these are the basic things about sexual reproduction. Now, on the basis of gametes, this sexual reproduction can be classified as, so these are the types or basic classification. It can be called isogamous type of sexual reproduction, where all gametes are identical in all respects, that is structure, function. So, morphologically, physiologically they are exactly same. So, then if these two gametes, for example, these are two gametes, both are spherical, same size, same physiology, everything same, and they fuse to form a zygote, then because the gametes are exactly identical, that sexual reproduction will be called isogamous type. Example, monocystis. If the gametes are not identical, one is bigger, other is smaller, or one has a different structure, the other has a different structure, then that would be called anisogamous. In case of isogamous, the gametes are identical in all respects, morphologically and physiologically. Here the gametes differ in morphology and physiology also. So, suppose this is the egg and the sperm is this kind of structure. So the shape is also different, the physiology is also different. Egg has cytoplasm which is going to participate in the zygote formation. Sperm does not bring the cytoplasm, so they are different. Example, all higher organisms like animals, including human beings, an isogamous type of sexual reproduction takes place. Third is called hologamous. Though it is called hologamous, it is nothing to do with whole, it is the whole, W-H-O-L-E, hologamous. Here the organisms, haploid organisms. So these are haploid organisms. They lead their life as haploid cells. This is seen in case of yeast. Now when they have to reproduce asexually, they reproduce by budding. But when they have to reproduce sexually, the two individuals, because they have been leading their life as haploid cells, these two haploid individuals start behaving like gametes. So basically it is fusion of two individuals, and that is why it is known as hologamy. And it will form a diploid zygote, and this zygote will undergo meiosis to form 4 haploid cells, which will again lead their life as haploid cells. So basically here it is fusion of haploid organisms. That is why it is called hologamy. So this classification is based on the types of gametes. If they are identical, isogamous; if they are non-identical, aniso; and if the whole organism is fusing, then it is called hologamous type. Then in case of sexual reproduction, the fusion of two gametes is known as fertilization or syngamy. Syngamy is fusion of two gametes, which is also known as fertilization. So syngamy or fertilization. This fertilization can also be of two types: self-fertilization and cross-fertilization. And cross. Self-fertilization is when the gametes of the same organism, they fuse with each other. This is seen in case of some plants. In plants, the flowers are bisexual. They have the ovary part also, which produces the egg, and they have anthers which produce the pollen grains. Normally they favor cross fertilization. The pollens will get transferred from one flower to another flower of another plant. But if that fails, then how to continue the race? Then they have this possibility of self-fertilization also. That means the pollen grain of the same flower can fall on the stigma of the same flower, or the pollen of a flower of one plant can fall on the stigma of the flower of the same plant. This is self-fertilization. Cross-fertilization, that means the male gamete from one individual goes to the female gamete of another individual. Cross-fertilization normally requires transfer of the gametes. So this classification is based on which type of gametes are fusing. Are they coming from the same individual, or are they coming from different individuals or two separate individuals? So syngamy or fertilization. Normally when we talk of sexual reproduction, we are talking of syngamy or fertilization. But there is a preliminary or a very primitive way of sexual reproduction which is known as conjugation. Conjugation is not exactly a method of reproduction. It is a method of genetic variation. So in conjugation, what happens is, two organisms, suppose we talk of Spirogyra. So these filamentous alga, they come or align close to each other a cross bridge is formed, which is called the conjugation bridge. Some genetic material from the cells gets exchanged and this bridge breaks. So, after the bridge breaks, these two filaments, they separate. But if this one had, say, genes A, B, C, and this one had genes 1, 2 and 3, there would be some exchange of genetic material which would take place. Say A from this goes to, say this is filament A and this is B. A from filament A is transferred to B. So, now B has A and its own 2 and 3. Whereas this filament got 1 from filament B, so it has 1 and its own B and C. So, there is a genetic recombination which has taken place. After they separate, they will reproduce by their own method of reproduction, maybe spore formation. So, conjugation is considered as a primitive method of sexual reproduction. The zygote which is formed, the zygote which is formed by the fusion of two gametes, male gamete and a female gamete. Helps in regaining the species specific chromosome number. It is diploid, and then this zygote undergoes mitotic divisions to form an organism. So, this is basic. Why we are just discussing the basics of sexual reproduction? Because the next two chapters of this unit are the details of sexual reproduction in flowering plants and then sexual reproduction in humans. So, anyways, we have to talk about all these things in detail. That is why just a brief introduction of sexual reproduction. One more, rather two more important terms related to asexual and sexual reproduction. Asexual reproduction is also known as blastogenesis. And sexual reproduction, where fusion of gametes takes place and zygote is formed, is known as embryogenesis. So, these two terms are also used when we are talking of sexual reproduction and asexual reproduction. Some organisms show a different method of reproduction. So, let us talk about parthenogenesis now. Parthenogenesis basically means that the egg gives rise to an organism without fertilization. This process involves formation of a complete organism from an egg without fertilization. Eggs are haploid. They attain that 2n number of chromosome after the male gamete has fused with the egg or it has fertilized the egg. But if egg is not undergoing fertilization and just starts dividing to form a complete organism, then that process is parthenogenesis, and the organism which is formed will also be haploid. Some organisms show this method of reproduction naturally. So, there is natural parthenogenesis and artificial. Natural means some organisms which show this method on their own, and artificial is when human beings or we stimulate the egg to undergo division without fertilization has taken place. So, artificially also such type of organisms can be obtained, and there are some plants, some organisms where even naturally these kind of reproduction is taking place. Let us talk about two scientists. Natural parthenogenesis was explained in insects or aphids by, aphids are insects, by Charles Bonnet. Charles Bonnet for the first time explained the process of natural parthenogenesis in aphids, which are insects. This term parthenogenesis was given by, the term was given by Richard Owen. He just gave the term parthenogenesis, but the first time whether this type of reproduction takes place or not, that was given by Charles Bonnet. Let us talk about these natural methods because they are occurring in nature without human intervention. Let us see how these organisms reproduce. So, we are talking about the methods of natural parthenogenesis, and then we will come to the artificial one. There are three different types in which natural parthenogenesis takes place. One is known as arhenotoky. Arhenotoky, in which only males are produced by parthenogenesis, or only males develop by parthenogenesis. We see this in honeybees. The drones or the males, they develop by parthenogenesis. So, in honeybee, that is Apis species, Apis indica and all, in some parasitic wasps and in mites. So, in these organisms, the males develop by parthenogenesis. What happens in them? The queen lays two types of eggs, one which have been fertilized and ones which are unfertilized. The treatment which is given to these cells is different. The fertilized eggs are also treated in different ways. Some fertilized eggs are fed with royal jelly and some are fed with bee bread. The eggs which are fed with royal jelly, they will develop into queens, and the ones which are fed with bee bread, they would develop into workers. Both these are diploid because these are coming from the fertilized eggs. Only thing is, queen is fertile, female, and the workers are diploid but sterile, females. So, both these which are coming from fertilized eggs or which are formed from fertilized eggs, they are all females. They can be fertile or sterile depending upon what are they fed with. The unfertilized eggs which are haploid, they develop into drones, and drones are males. They are haploid males. So, all the males which are found in the colony of a bee or these parasitic wasps, they are all haploid males coming from unfertilized egg. So, this is egg which has given rise to a complete organism without fertilization. So, when we talk of arhenotoky, it is only males which develop by this method of parthenogenesis. The second method which is naturally seen is known as thelitoky. In thelitoky, only females develop by this method of parthenogenesis. So, here only females develop by parthenogenesis. This thelitoky is also known as complete parthenogenesis. And arenotoky is known as incomplete. So, this is one more term given to it. In thelitoky, only females are found in the colony or in the complete species which exist. There are two examples where we find this very commonly, naturally occurring. One example is Caucasian rock lizard. Okay, I am going to write these examples here. So, these are Caucasian rock lizards. Scientific name of this Caucasian rock lizard is Lacerta saxicola armeniaca. This is one example where there is a female. This female lays eggs and this egg develops into female without fertilization. So, where is the male? Why is the male required then? So, there is no male required. So, in this complete species, there are only females. So, it is a female which lays eggs. The eggs are haploid and they again grow into females. So, the females are haploid. They lay haploid eggs. These haploid eggs without fertilization again produce females. The only negative is there is no genetic variation. Genetic variation is when this combination of gametes would take place. So, this is one example. The other example is of Typhlina brahmina. It is the smallest Indian snake. So, these two examples are the examples of thelitoky. So, let us connect this so that. So, examples of thelitoky or complete parthenogenesis. Now, it is called complete because only one type of sexual organism, only females are there, males are not even produced, they are not even required there. So, this is thelitoky. Two examples where we see it is Lacerta, which is Caucasian rock lizard, and Typhlina, which is a type of snake. So, either males produced or only females produced, two types. The third is known as amphitoky. Amphitoky is known as cyclic parthenogenesis. Arenotoky was called incomplete, thelitoky is known as complete, and amphitoky is known as cyclic parthenogenesis. It is seen in aphids. Aphids, again insects, in which Charles Bonnet actually saw this natural parthenogenesis. So, in aphids, both males and females can develop by parthenogenesis. It is just generation by generation. So, in one generation haploid eggs produce males, next generation haploid eggs will produce the female. So that is why it is called cyclic. So both males and females can be developed by parthenogenesis. Both males and females develop by parthenogenesis. So these are three naturally occurring ways or methods of parthenogenesis. Arenotoky, where only males develop by parthenogenesis, which we see most commonly in bees and wasps. In case of thelytoky, only females. There are two very important examples, and amphitoky, which is seen in case of aphids, which is cyclic. So alternatively, both sexes can develop from it. These are natural methods. Artificially, we can trigger the cell to undergo division. So there are some factors or stimulants which can be used. So artificial can be done by chemical or physical stimuli. There can be some chemicals. Very simple chemicals can also trigger parthenogenesis, and this is normally seen in lower organisms. Up till frog and all fishes, frog, this is seen. Chemicals like ethanol or organic solvents, saline, calcium chloride, chilled calcium chloride can stimulate, but it depends on the different species. Physical things, electric shock, temperature variations, sometimes even a prick of a needle can also trigger this division. So prick of a needle. In case of frog, a needle which was dipped in blood, when that was used to trigger the egg of the frog, it started division and it gave rise to an organism without fertilization. So that was in frog. So in frog, example, needle dipped in blood. When was used to prick the egg underwent parthenogenesis. So artificially also we can trigger this process, but naturally also there are certain organisms which show this process. So parthenogenesis is a very unique kind of reproduction which is reported in organisms. So now we have seen all three methods: asexual reproduction, sexual reproduction, though in brief, and parthenogenesis. Now we are talking about reproduction, only one stage of the life. So we need to understand which stage are we talking about. What are the other stages? So let us just take the stages of the life.